So now we move further. Now we talk about the chromosomes that are carriers of the genes. So chromosomes, we know that all species have a fixed number of chromosomes. However, the characteristics of species, including physical appearance, body functions, behavior, etc., are not simply outcome of chromosome number, but these are the result of units called genes, which the chromosomes carry. The lion and the cat have the same number of chromosomes, 38, yet one is distinct from the other in body size, appearance, color, behavior, etc. All such characteristics of an organism are the result of the genes located on the chromosome. So what we see is that one thing is, of course, we saw till now was the chromosomes. Chromosome number ke karan, okay, we are 46, that's why we are humans. But yeah, this is not the only characteristics. The characteristics what we are getting is also because the chromosomes जो अंदर है, chromosomes के अंदर है genes. Now the genes which are there in the chromosomes, that is the genes, that is this particular genetic ka genes, is the one which is going to be giving that particular characteristics to that organism. Like they give you the example over here that cat and lion both have 38 chromosome sets. Okay, so 38 chromosomes. So if they both are 38, why one is a small baby cat and the other one is a huge lion? So this is basically because the gene which the 38 carried in the lion are different from the 38 which are carried in the cat. So accordingly, we have this new kind of definition, and that is of the gene. So we have over here that genes are the specific parts, DNA segments of the chromosome which determine the hereditary characteristics. So DNA has got certain kind of genetic material in it. So that genetic material which is present in the DNA is going to identify the hereditary characteristics which are going to be going from one generation to another generation. So that is what is genes. The number of genes in human, according to the latest finding, there are nearly 30,000 genes in humans. Chromosome number 1 has the largest number of genes, that is 2968, and the chromosome Y has the fewest, that is 231. So you can see that each and every chromosome has got multiple genes in them. And that multiple genes are going to give you the different characteristics. So accordingly you can see that what are the different kinds of features which can be changed, which can change. And that is also one of the reasons that when you walk from one place to another place, suppose in a crowded place you are moving around, you can see that each and every person is different. Why is it so many difference in each and every person when there are only combination of 46 chromosomes? No, it is not the combination of 46 chromosomes, it is the number of genes which are present in the chromosome which make each and every person completely different from the other. You are not getting confused. Kerry, I talked to you and I know, you easily come to know. Then I didn't speak to you but I spoke to somebody else. So that's why there is no confusion about you particular seeing them. You cannot get confused that you cannot get confused unless and until they are identical twins. There are very few chances that there will be some resemblance, but you will never find that somebody is exactly the same. You can easily identify, okay, this is Shah Rukh Khan and this is Amir Khan. You cannot get mixed up between the two. Okay, so this is because of the number of genes which are present in the chromosomes, which is giving you a large number of different kinds of complex combinations possible, which is giving you an individual which is completely different from the other individual. Hence, the genome is the full complement of DNA, including all genes and the intergenic regions of an organism. So accordingly, जितने भी genes हमारे body में present है, all that complete gene is a complete set of DNA. That full complete set of DNA is called as a genome. So it is genome. So genome is the complete set of the full complement of the DNA, including all genes and intergenic regions. So There is also some intergenic regions between the two genes. So connecting genes is the intergenic regions. Now we see the next part, and that is the genes and their alleles. Now genes is, as I said, is a particular kind of the specific DNA segment. Now this DNA segment which is there, it will be having a possibility that it will be either one particular trait or another particular trait. So according to the trait what that gene is following, it will be giving rise to a thing which is called as allele. So what you see is normally every gene has two alternative forms. So all the genes will have two, like I said, left-handed, right-handed, tongue rolling, tongue non-rolling. We say each and every gene, each and every character what we are having will be having two alternative forms for the character producing different effects. These alternative forms are called as alleles. So the alternative forms of the gene. So gene ka alternative forms are the alleles. For example, in the character tongue rolling, projecting the tongue out of the mouth in the form of tube, there are two possibilities. Either one can roll the tongue or one cannot roll the tongue. It will never happen. नहीं उसका आधा tongue roll हो रहा है, दूसरा नहीं हो रहा है. So not possible. It is going to be either tongue rolling or non-tongue rolling. So tongue rolling रहेगा तो it will be like this, and non-tongue rolling रहेगा तो वो tongue बाहर निकलेगा तो भी flat ही बाहर निकलेगा, वो roll कर ही नहीं पाएगा. It will be just like this. So that is non-tongue rolling and tongue rolling. So you see over here that thus there are two alleles in this gene. One is tongue rolling and other is non-tongue rolling. So over here it is being seen that the one of the two traits, that is either tongue rolling or non-tongue rolling, will be dominant. Okay, that means maximum times that will be the one which is going to overcome the other one. See, because if I am having two kinds of genes in me, okay, one is a left-handed, other is right-handed, then which will I be inheriting? Whether it's a left-handed or right-handed, then it will depend upon natural factor, that naturally it is always the right-handed which is dominant than the left-handed. Hence, even if I have got both the genes, one from my parent as left-handed and one from my parent as right-handed, I will be showing the property of right-handedness and not the left-handedness. So that is why out of the two alleles which is possible, one of them will be dominant and other one will be recessive. So one dominant, other is recessive. So we see out of the two alleles of the gene of a gene, one is dominant, that is super ruling, and the other is recessive, that is subordinate or submissive. Okay, so one is dominant, other is recessive. To understand this again, take the example of tongue rolling. A large number of people can roll their tongue into a tube projecting out of the mouth. Try whether you can do it or not. Ask your classmate to try it. Most of them can do it. You will find that in a classroom where there are maybe 100 students, in that out of 100 students, 75% of the students will definitely be tongue rolling. And only 25% will be such, or even less than that will be such, who cannot roll their tongue. So you can see the gene of tongue rolling is located on a particular pair of chromosomes. Thus every individual has a pair of these genes. It is a rule in genetics to represent the dominant gene as capital letter. So if you are talking about tongue rolling, we are taken as a capital R. So the non-tongue will be, so this is rolling and this is non-rolling. So if it is tongue rolling, it will be capital R. Non-tongue rolling will be shown as small r. So these are the two alleles of the chromosome which is going to have the characteristics of tongue rolling on it. So thus every individual has a pair of this gene. It is a rule in genetics to represent the dominant gene as a capital letter. So here R for tongue rolling, and the recessive gene as the same but small letter like small r for non-tongue rolling. Thus a recessive gene is one which is in the presence of contrasting dominant gene is not expressed. So if a particular kind of organism has this both. Okay, of course everybody will be having it in pair. So अगर किसी के पास इस तरह से pair में है तो this person will be tongue rolling. So we can see now the example over here. So then what are the possibilities of this pairing? It could be that both of them are RR, that both are capital R, then there is definitely rolling. It could be that one is small, big R and the other is small r, or it's a small r or in a big R. Suppose we are talking about this is the mother and this is the father, or male and this is female, this is male and female, this is male and female. So the father and mother, or father mother कर देते हैं इसको. तो father मुझे mother से capital आया, father से capital आया, या तो I am definitely rolling. मदर से कैपिटल आया, फादर से स्मॉल आ रहा है, या मदर से स्मॉल आ रहा है, फादर से बिगर आया, तो भी आई एम टंग रोलिंग ओनली. But if both mother and father both give me smaller, only then I am non-tongue rolling. Got it? So that's why the presence of even one single capital R in the chromosome automatically gives him that particular characteristics as the dominant characteristics. Okay? So you see over here that a recessive gene is one which is present in the presence of contrasting is not expressed. So this and this does not express themselves, hence they are called as recessive kind of allele. So alleles can be defined as alternative forms of gene occupying the same position on the homologous chromosome and affecting the same characteristics but in different ways, because they have two different beats. One is dominant, other is recessive. So it is the form of a gene which is present in the same chromosome, occupying the same location, but it is going to be affecting the same characteristic but in different ways. The pair of chromosomes showing 4 imaginary genes A, B, C, D, and the situation depletes here is capital A is homozygous. So you can see over here, this thing can be given a particular kind of name to the pairs of chromosomes. So over here, if this is the one that is a capital R with a capital R, they are called as homozygous dominant. Okay? Homozygous. Homozygous means same. Zygous means like forming the zygote. So if it is formed by the same kind, it is homozygous. And because it is giving a dominant characteristics, that is why it is called as homozygous dominant. Okay? If it is capital R with a small r or a small r with a capital R, then these two are called as heterozygous. It is heterozygous. Now the heterozygous, but even if it is heterozygous, we always see that it will be the dominant characteristics which is going to be shown as a character. So both of them are tongue rolling. That's why it is still dominant, because it is what is going to be given out will be the dominant characteristic. It is this which is going to be super ruling the small r. So that's why these are called as heterozygous dominant. And if it is smaller, then that is called as, of course, the same. So again, homozygous. But these are recessive in nature. That's why it is recessive. So it is homozygous recessive. So if a person is non-tongue rolling, he has got the chromosome type as homozygous recessive. But if somebody is tongue rolling, you cannot identify whether it is homozygous dominant or heterozygous dominant. Okay. So accordingly, you have that capital A is homozygous dominant pair, capital B, small b is homozygous recessive pair, and capital C and small c and capital small d and capital D are heterozygous dissimilar pairs. So the chromosomes as a whole are neither dominant or recessive. As a whole, the chromosome is not dominant or recessive. It is the allele present which is dominant or recessive. This is one part. This is not a dominant get. It's always a pair. So we do not call a chromosome to be dominant or recessive. Accordingly, what we see over here that we are having the two things. One thing is the type of gene what it is having. So one is type of gene, that is homozygous dominant or heterozygous dominant or homozygous recessive. That is the type of gene. This type of gene is called as genotype. And second thing is, it tells me whether it is rolling. So this one we know that these two are rolling, and this is non-rolling. So we can see that the type of character or the physical character shown by them are either rolling and tongue rolling. So that is the physical character. So the next one is type of physical character shown. So what is the physical character shown? That is called as a phenotype. So over here, if I talk the genotype over here, the genotype are RR, R smaller, bigger, smaller. These are the genotypes. Over here, what are the phenotypes? Rolling, non-tongue rolling, non-rolling. Understood? So, if I talk about the genes, the type of gene that is having, the kind of the gene it is having over here, then it is called as genotype. And if I talk about the type of the characteristic it is showing, that whether it is a tongue rolling or non-tongue rolling, it is called as the phenotype. So hence we see over here, the three situations pertaining to any pair of genes are as follows. In case of tongue rolling is capital R, that is dominant, he is a tongue roller. Capital R, small r, capital R, that is one dominant, one recessive, he is still a tongue roller. And the third one is both small r, that will be a recessive one, he will be a non-tongue roller. You can see from the above example that the tongue roller are of two kinds, that is genetically constitution of capital R or capital R small r. So both one, this is capital R and capital R small r, both are tongue rollers, whereas non-tongue rollers have only one genetic constitution, that is smaller. The genetic constitution pertaining to the kinds of genes possessed are called as genotype, and the expressed shown characters are called as phenotype. Thus, for a phenotype, for tongue rollers, there are two genotypes, that is homozygous dominant is one genotype, heterozygous dominant is one genotype for tongue rollers. So tongue rollers has got two kind of genotypes, that is homozygous dominant, heterozygous dominant. So homozygous dominant, that are two, both pairs are capital R, and heterozygous dominant with dissimilar pairs, that is smaller, bigger, smaller. Okay. And the genotype of the non-tongue roller is just one single homozygous recessive. So genotype for rollers are 1 and 2, and that for a non-tongue roller will be just 1. So whereas you can see, so we can define the genotype as the set of genes present in the cell of an organism. The set of genes present in the cell of an organism is the genotype, and phenotype is the observable characteristic, whether tongue roller or non-tongue roller, rolling or non-tongue rolling. So that is the observable characteristic which genetically controlled are called as the phenotype. So that is how we are having the different kinds of allele present in the chromosome, in the gene, sorry, not the chromosome, in the gene, and this gives rise to the homozygous dominant or heterozygous dominant or homozygous recessive, giving rise to either tongue rolling or non-tongue rolling or any such characteristics which we learn by our different genes, or we got from our different genes. So that was one part. Now we come to the next part, and that is called as a pedigree chart. So when you are talking about a family and you want to find out that what are the different kind of recessive and dominant kind of nature of the different members of the family, then we make a chart which is called as a pedigree chart. Now in a pedigree chart, there are going to be four kind of symbols. One will be a open circle. This represents a female. One which is a square which represents a male. Now the female, if the female over here is shaded over here, then he or she becomes, sorry, not he or she, it is the female becomes a recessive female. So if I show a shading over here to any particular circle, then it means that that particular female is a recessive trait. That means, suppose we are talking about tongue rolling, then if I shade a female over here like this, means she is a non-tongue roller. Okay. Similarly, if I am going to shade a square, then he is a recessive male. So recessive male will be represented as a square like this. So accordingly, if we see a family, the family will be this way, that this is a family having a mother and a father. So let's consider tongue rolling as the example itself. So this is the mother, this is the mother, this is the father. So the mother and father are over here, and they are both non-shaded. That means these two are tongue rolling. Okay, this she is tongue rolling and he is also tongue rolling. Now they have got kids. So let's consider they have got 3 kids. So one is a girl who is non-tongue roller. Then there is a boy who is a tongue roller, and is a girl who is also a tongue roller. Okay, so you can see over here, this particular thing tells me that how is the differentiation taking place between the family wherein the mother is a tongue roller, father is a tongue roller, but the 3 kids are not identical. 2 of them, one is a girl who is a tongue roller, boy who is a tongue roller, but the girl is a non-tongue roller. When you try to understand this thing, we can easily also represent that what will be the kind of gene, which is genotype. See here, what we are showing over here is the phenotype. The phenotype is tongue rolling, non-tongue rolling, non-tongue rolling, tongue rolling, tongue rolling. This is what we are showing as the genotype, a phenotype. But if you talk about genotype, that is RR, small r, big R, wala, तो उसमें मुझे ये मालूम पड़ता है कि this is definitely small r, small r. Now, when can this be smaller, smaller? If both of them contain one smaller in each of them, only then it is possible. So, accordingly, this will be smaller, bigger in both the cases. This we cannot identify. They can be both smaller, both bigger, or one small, one, or it could be the same. So, it could be even this way in any case. So, it will be this way also. Yeah. So, we cannot identify this one and this one, but we can easily identify this two and this if one of them is a non-tongue roller. So, yes, you can see the illustrates a family. So, from parent to child, tongue rolling, an example of inheritance. It is a family chart of 2 parents and their 3 children. Again, illustrate the trait of tongue rolling. So, you can see over here in the pedigree chart, the inheritance is the ability to roll the tongue. So, the figure O is a female, square is a male, solid one is a recessive one, and this is a recessive male. So observe the following points. Male are shown in squares and females in circle. Both father and mother are tongue rollers. They are hollow, so they are tongue rollers, represented by usual express characters. And on the 3 kids, 2 are rollers, whereas 1 cannot roll. So that's why 1 is solid. So that is what is being shown over here. We can identify the recessive trait smaller of non-tongue rolling is in one of the children could have come from nowhere else but the parents. So this has to come from the parents only. So the non-tongue roller child in the family chart with genotype RR must be received 1 R, that is small r gene, from one parent and the other smaller from another parent. That's why they both are having smaller over here. Conclusion is that each of the 2 parent is heterozygous dominant. So these both are having heterozygous dominant kind of genotype. So if one, on the other hand, one parent was homozygous then and dominant, the other is heterozygous, dominant, then every child would have been at least one dominant. So in case, suppose if I say that ये possible नहीं है, ऐसा होता, अगर ये ऐसा होता, तो definitely what would happen? Even this child would get at least 1 hour from the parent. Then would it be non-controlling? No, not possible at all. Okay, so here the presence of a recessive trait here tells us that these have to be recessive, these have to be heterozygous. So we can see that if on the other hand, one parent was homozygous dominant and the other heterozygous dominant, then every child would have got at least one dominant gene from the homozygous dominant parent through the sex cell. The situation can be schematically explained, which is given in a Punnett square. So next what we are going to learn is a Punnett square. Okay.